I'm Dan Kastner, and I'm at the NIH. And I had the opportunity, the real honor and pleasure of moderating the session today. And it really has been just an absolutely wonderful, wonderful meeting. And we have to thank Chip Chambers and the others in the organizing committee. Chip is going to be coming out once I've given my brief summary to tell you a little bit more, but certainly he has been absolutely wonderful in terms of putting this all together, organizing it, raising the funds for it, getting enormous numbers of people here on very short notice, with somewhere around 150 healthcare professionals and 60 or whatever the number is of patients and families. It's really been incredible, and incredible when you think about the fact that this is a meeting that was really only discussed on a concrete basis beginning about in the end of February, February 29th, being Leap Day of this year. So we've had 4 major sessions during the course of the day today. And we've talked about 4 different topics with each of those sessions. And they really underscore the fact that we have learned so much in the course of the last 2.5 years since the initial publication of the 2 papers describing this condition, DADA2. And so I'll just summarize for you a little bit about what was discussed there. Of course, the initial recognition of DADA2 as a clinical entity was focused on either the stroke manifestations of the disease or on the polyarteritis nodosa manifestations, that is, the inflammation of the blood vessels. And over the course of the last 2.5 years, we've really gotten a much broader understanding of what mutations in this gene can do in terms of pathology in humans. And so, for example, we've learned that there can be problems with the liver in patients who have DADA2, that there can be problems with immune deficiency, with having increased susceptibility to infection, and that certain cells of the immune system, which we hadn't really thought were affected by this condition, can be. We've also learned that at least in some patients, there can be a problem with the red blood cell development, and that there can be problems sometimes with a certain type of large granular lymphocyte that can sometimes proliferate more than it should. There are a couple of other conditions that sometimes are seen associated with mutations in this gene, something called Sneddon syndrome, which is actually a condition where adults sometimes can have the rash that's associated with DADA2, the livido rash associated with DADA2, and can have strokes, and perhaps something called Castleman syndrome as well, in which patients get enlargement of the lymph glands. So this is simply to say that what we had thought was DADA2, it turns out that there are a lot more conditions that are associated with mutations in this gene. In telling you about this, I'm not in any way trying to imply that these particular manifestations are necessarily going to affect anyone who is here who doesn't already have those conditions. But it is to say that, number one, it alerts us as physicians to be on the lookout for mutations in this gene in patients who have those particular conditions, because we can then make a diagnosis in those individuals. And as I'll get to in a little bit, there are very promising treatments that are now available. Again, information that has come about really in the last 2.5 years, which is then available for those individuals. And that really is terrific as well. There's a lot more that we need to do in order to understand this disease, and so I'm not saying that in the last 2.5 years we've learned everything that we need to learn in terms of the disease, and certainly one of the things that we're going to need to do is to define whether or not there may be individuals who are carriers for the mutation who might have some symptoms. What happens to individuals who are asymptomatic, who test positive for 2 mutations in the gene? What is their risk of having later on some manifestations? But this is the kind of thing that, because we have a group of people together, physicians and patients and families who are very, very interested in this condition, we can actually then make plans to answer some of those questions by doing careful studies, following the clinical course of patients, and learning what is really the natural history of the disease. And in order for us really to understand what would be the best treatments for any disease, we have to have a good idea of what is the baseline that we're trying to deal with. And so this is extremely positive, both in terms of what's been done in the last 2.5 years, and the fact that we have really a group of very motivated people together who can help us to answer the questions that still remain. And by having everyone together, we've defined some of those questions that we need to address in the course of the next year or two. So that really is very, very important. So that was the first session, was dealing with sort of defining the disease. The second session had to do with screening people, making the diagnosis, figuring out who has DADA2 and who doesn't have DADA2. And of course, there are different ways that one can approach that question. One of the ways that, of course, has been used now since the description of the disease has been DNA sequencing. And a second way that also has been available since the get-go has been the measurement of levels of ADA2 in the blood. And certainly we've seen some great advances in both of those areas as well, so that over the course of the last 2.5 years, we now know many, many more misspellings of the gene, mutations, whatever you want to call them, that are associated with the condition, and just understanding what the landscape is of course extremely important. There have been advances in terms of the protein assays, so that now, rather than having to have some liquid blood, one can actually make the diagnosis from a spot of plasma on a piece of filter paper. And so that's actually a big advance in terms of our being able to move rapidly with this. We identified some things that we definitely need to work on, such as standardization of these studies and getting what's called CLIA certification for these studies. And there was a very, very lively discussion with regard to who exactly should be screened for mutations in this gene. Who should be screened for DADA2? And certainly some of the patient groups that one could say would probably be high on the list would be individuals who have had unexplained strokes, individuals who have had problems with their liver, with portal hypertension, individuals who have immunodeficiency, individuals who have problems with bone marrow development. These are all conditions that have been associated with DADA2, and certainly one could make the argument that undiagnosed individuals with any of those conditions probably should be screened for mutations in this gene. And we had a discussion of, well, should we actually institute screening in newborns for this condition? Because at least some of the cases that we heard about today presented very, very early in life. And related to that is the question of, well, why is it that some people present very, very early in life, within the first couple of months of life, and other people don't have any symptoms until they're 10 years old or whatever. What's the difference there? Why is that? Is that genes? Is that some sort of environmental effect? And again, having a very systematized, careful study of patients who have this condition will allow us to figure out some of those questions so that, for example, could it be that infections are triggering the exacerbations of the disease? These are very important things for us to understand and very important things for you guys to know about just in terms of thinking about your own health and anticipating when problems might ensue. So that was the second session. So the first session, defining the disease. The second session, screening for the disease. The third session had to do with pathophysiology, and that was really fantastic. It was a great session, and there we learned a lot of new things about the biology of ADA2, the protein that's abnormal in this disease. And so we learned more about the proposed pathophysiology of this disease, that ADA2 appears to be a growth factor for certain kinds of white blood cells and perhaps for the lining of the blood vessels as well, and learned more details with regard to that process. But there were some other things that were really cool that we heard about too. And so one of them was just the idea that probably adenosine does play an important role in the pathophysiology of this condition, that as some of you may know, ADA2 is an enzyme that can convert adenosine to inosine, and perhaps the inability to do that outside of the cells really is an important part of the pathophysiology of that. Why is that important? Well, it's important because that would allow us, perhaps understanding that, to eventually develop new treatments for the disease. There was also speculation and some data to support it that in fact interleukin-10, which is one of the cytokines that's involved in regulation of the immune response, might have something to do with the regulation of ADA2. And that would be a very important thing as well. A third thing that we learned that I thought was extremely interesting was the idea that ADA2 may in fact regulate through a fairly complicated pathway, but may regulate the production of interferon. And the reason why that's important is that there are actually drugs, FDA-approved drugs, on the market right now that would have an effect on that pathway. And so that gives us some ideas in terms of additional therapies that might be available for patients. And so really, understanding the pathophysiology of a disease is so important in terms of developing treatments. There's always the example of serendipity where you just stumble on something and you find a really good treatment for a particular disease. But if you understand what's going on, you're in a lot better position to be able to say, well, we think that this or that kind of medication is going to be beneficial for patients. And so that really was a very important session. And then the fourth session was the session on treatment of DADA2. And it also was spectacular. And there were fireworks going off in this room before you guys came in. We had to, sort of sweep off the carpets because there were cinders on the rug from all the pyrotechnics that were going on here. So we had a discussion about TNF-Inhibitors as one of the treatments for DADA2, and certainly in patients, at least with certain forms of DADA2, particularly those patients that have the vasculitis form of DADA2, which was first really recognized, as a treatment in Israel a number of years ago, TNF inhibitors for that form of the disease, and the stroke manifestations of the disease. TNF inhibitors seem to be pretty effective, and we really do need to collect together all of the data from everyone so that we can get an idea of exactly how effective they are, but we heard really encouraging data with regard to that. We also heard very encouraging data with regard to bone marrow transplantation for patients that have the bone marrow failure manifestations of DADA2. And we had a very lively discussion, that's where the pyrotechnics came in, with regard to when would one offer a bone marrow transplant for a patient, and what is the risk-benefit ratio? These are really, really important questions, and for you guys to think about those kinds of options, one has to have good data to make intelligent decisions, and that's where getting everybody together and pooling together everyone's experiences is really important in terms of making these kinds of really very, very important decisions. A couple of other things that were discussed as potential treatments included gene therapy, and certainly that's a very active area of research right now, and also a modified form of ADA2 that's what's called pegylated, so that it lasts longer in the circulation. And so one of the things that we resolved to do at the end of this meeting is really to gather together all of the data that we have with regard to responses to treatment amongst all of the people at all of the centers that are now being followed for DADA2 and treated for DADA2, so that we can really get some good comparative data. But the data that we heard, I will say, was extremely encouraging. And when you realize that this was a condition that 2.5 years ago, speaking as someone who was involved in addressing this condition 2.5 years ago, although it was very exciting to have recognized a new disease, it was also very daunting just thinking about, well, what are we going to do for patients with this condition? Because at that point, we didn't know. And so the idea that 2.5 years later, we actually have some really good options is really, really incredible. And I think that it's a testimony to the hard work of all of the people in this field and to people like Chip Chambers for getting everyone together to talk about it. And so with that, I think I'll end the summary here. Those were really the 4 major areas that we talked about.